Ladies and gentlemen, it's good to be with you. I am Dr. J. Frederick Templeman, and this is Dr. Ramita Watanapokasan from Thailand. We're going to be talking to you today about research with the mangosteen. But before we begin, I think it is very important for you to understand that we will be talking about research which is done in vitro, meaning in glass literally, and that talks about the growth of human cancer cells in a petri dish. We will also be talking about in vivo animal studies with cancers, human cancers. But what is important to take away is that the information we will be talking about cannot be used as a basis for concluding that the same observations we can see in vitro or in animal studies can necessarily occur in human beings. It would be wrong to conclude that this research has any direct relevance on human cancer, except that we can conclude that there may be qualitative benefit from the use of the mangosteen as a dietary supplement. We cannot talk of it in positive or in absolute terms because there is no quantitative benefit that has been proven. The trials have simply not taken place. It is also important to understand that if you are excited about what you hear today, it would be normal that you would want to get more information. The studies that Dr. Watanapokasan will be putting out will be written and published within the next few months. At that point in time, we will make certain that you can find the journals in which these articles will be published and read them for yourself if your interest goes that deeply. What is important is that you do not attempt to contact the universities where she has done her studies, nor if you have received previous information regarding the content of her studies, do you want to make that public because it would prejudice her ability to publish original material. We do not want you to contact the universities because we are now beginning to obtain some of the respect from very important and prestigious academic institutions which will allow us to push the research for the mangosteen much more fervently than we have in the past. If a large number of people were to contact any university involved in this study and were to pester them, and you'll excuse me for using that term, to get the information, it would irreparably damage our relationships with academia and make it certain that we would set back the program of research into this wonderful food supplement for many, many years. So please, do not take more from this information, which is very satisfying and very important by itself, and do not contact the universities involved. Thank you. Now, I'm going to be asking Dr. Watanapokasan a number of questions that I hope will allow us to understand more fully what she has been doing and what the results have been. Dr. Watanapokasan, you have been in the United States for the past year?
Yes.
And you were doing research on the mangosteen fruit, is that correct?
Yes.
Were you using the whole fruit or an extract, or what were you using in your studies?
I used the mixed xanthone.
So mixed xanthones.
Yeah, and also the alpha mangosteen.
And alpha mangosteen, which is one of the xanthones which was particularly extracted and used additionally.
Yep.
So you were using the elements from the plant known as xanthones,
yep.
and you were using them in both in vitro studies and with animals?
Yes, but I concentrate on the mixed xanthone in the animal.
In the animals?
Yep.
Okay, so although you did some in vitro studies, the major part of your study during this year was with animals.
Yeah.
Now, could you explain to us why, when we are investigating the medicinal potential of plants, that we use animals first?
Oh, because after we get the in vitro results, so we have to step, to the animal. Because, for example, I use mice because
Yes.
they have the mice and men has about 85% of the DNA sequences that they have the 85% similarity.
So that in the species of mouse that you are using,
Yeah.
there is an overlap with
Yep.
human beings,
Yeah.
and 85% of the genetic material would be
We-
similar.
Yep.
And that, therefore, we can conclude that if we see certain results in mice, then there is a stronger probability that the same thing could be seen in humans.
Yes, that's correct. Yep.
Good. Now, in the studies that you did with the mice, what were you looking at?
I try to induce the tumor, human colon cancer in mice,
Okay.
and then after that I inject the mixed xanthone
Into the human...
into the tumor.
Okay. So
Yeah.
you would take a needle,
Yeah.
draw up some human cancer cells,
Yep.
colon cancer
Yeah.
cells in this instance,
Yeah, I grow up the human colon cancer cell
in a culture.
in the culture.
And then draw it up in a needle
Yep.
and inject it under the skin of the back
Yep.
of a little naked mouse.
Yep.
And then you would watch while the mouse incubated the tumor and
Yeah.
it began to grow.
Yeah.
Now, you had just one group of mice. How many groups of mice did you have?
At least 3 groups. Yeah.
At least 3 groups.
Yep.
Could you explain to us why you used more than one group of mice?
Yeah, because we have to make sure, like, okay, it really works. It really works.
So you have one group
One
of
group
mouse
of the control.
of a control.
Yeah.
And this means that the mouse is injected with the tumor cells,
yep.
the same as the others,
Yeah, the same.
but it does not get any intervention.
No.
Nature simply takes its course.
Yep.
Okay? And then in the test group of the mice, you inject the xanthones into the tumor itself.
Yep.
Now, what was the third group?
I did more than 3 groups because I have to vary the concentration of xanthones.
So you were attempting to see whether a small,
Yeah,
greater amount
yeah.
or the greatest amount
Yep.
would make a difference.
Yep.
In your observations, did the stronger dose of the xanthones work more effectively than the weaker dose, or was there effect at all doses?
Yeah, I mean, at the low dose, it has some effect at the beginning, and after that, the tumor start growing.
Again.
Yep.